Good afternoon. It's 3 o'clock precisely, 3 o'clock Central European time, and well, wherever you are. Thank you very much for joining this EFSA's webinar on the Novel Food Guidance Update. Well, on behalf of EFSA, we really want to welcome you to this session. The webinar should last about 3 hours, but I'll give some more details in the coming minutes. My name is Ana Afonso. I'm the head of the Nutrition and Innovation in Food Unit, who is responsible for the secretariat of the NDA panel, to which this work on the novel food guidance update has been put in charge. Next. Some housekeeping rules. You are now connected to the audio-video broadcast. Unfortunately, and because of the large number of participants, this is a one-way audio communication channel, and we cannot actively engage with you on conversation, but I'm sure we will have other opportunities. This event is held in English and is being recorded. The recording will be published as well on EFSA's website, along with all the presentations that we'll give today for future use and reference. After this event, you will also receive a link to a survey to evaluate how this event proceeds. Please fill it in. It's very, very welcome your feedback so we can improve and make it better for everybody. Okay, maybe not so fast. So we are super happy with the amount of registrations we received for this event. We have 844 registrations from 62 countries all around the globe. So this it makes us very proud that all of you are so interested in all the work that we've been conducting. A little bit about we asked during the registration to register yourself in the following categories, and you'll see that largely we have about 53% of our registrations come from private sector, and this is definitely a target audience that we are very much interested in engaging with, our future applicants on novel foods. Next. Okay, so what do we want to do with this webinar? We want to explain the main elements of what changed in this updated guidance document, so that this hopefully will also help you in being more clear about the public consultation. It's not replacing the public consultation by any means. It's to hopefully guide you on what kind of input and feedback we would like to receive, and actually to alert you for this public consultation so we can receive as many comments as possible. We will start to address questions and requests that were received during the survey registration for this webinar, but this does not stop that you will also make those comments during the public consultation. I already introduced myself. My colleagues, Andrea Germini, who's the team leader on the Novel Foods Product Characterization team, will be with us. Ermolaus Ververis, who's the scientific officer on the team of Andrea. Emanuela Turla, Senior Scientific Officer in the Novel Food Product Characterization, George Kass, the Team Leader of Novel Food Product Safety, and Wolfgang Gelman, Senior Scientific Officer in the Novel Food Product Safety team. There's far more of our staff involved in this work, and to all of them I wish to say a very big thank you, and also to my colleagues in the engagement team in EFSA that facilitate the organization of this and other events. So a little bit on our agenda. The first presentation will be by Andrea, and he will give you an overview of what is our work in the risk assessment of novel foods since 2018 in particular. We will then ask Ermolaus to give you an update on the novel food guidance, why we're doing, and what are the main changes. We will then have a little break, and I will ask you not to disconnect during this break to facilitate the technical part. And after that, what we will do is try and answer as many as possible of the questions we've received. This is the largest and probably the most important section of our agenda today. And then hopefully we'll just close the webinar with some indications on what are the next steps, what are the timelines, and so on. And I think this is probably all I need. So I would like to pass the floor to my colleague Andrea, and for the presentation about risk assessment of novel foods by EFSA. Andrea, the floor is yours. Thank you very much.
Thank you, Anna, and thank you to all the participants for being with us today. As Anna mentioned already, we have been, I will guide you through some of the recent work we have been doing on novel foods very briefly, because the core of the activities will be on the Q&A. But this is essential for us also to frame a bit the context of this guidance update. EFSA had already developed in the past, some years ago, in 2016, a guidance on the safety assessment of novel food. This one we are presenting for public consultation is actually an updated version of that guidance that goes way more into the details of the science we have been doing in the past years, and also take stock of the recent advances in the science and technology that has happened in the field. So it's not a totally new guidance. It's actually a fairly updated from scientific and technical level of details, but also with the addition of some new elements which are emerging in the area of novel foods, which we will tackle specifically later on. Briefly, if we go to this second slide, then what is novel food in the European Union? Because European Union has a specific definition which differs from other geographies of the world. In the EU, every food or ingredient that has not been consumed by human to a significant degree before 15th of May 1997 is considered as a novel food. This applies to several products and several categories of products. You'll see here a brief list of what recent products that have appeared on the market following authorization as a novel food. They are all classified into three large categories, basically new synthesized or isolated compounds, new sources of food, food derived from new processes and technologies, and an additional category introduced by the 2018 regulation, the new novel food regulation, which is traditional foods which have been consumed in other geographies for more than 25 years. The focus on this update, on this discussion today, is on the three first blocks, but we do have for public consultation also the updated guidance on traditional foods, which I would invite you to comment upon, together with the one on nutrient sources, which is strictly linked to the work we do. Okay, going to the next slide, then these are the categories defined by the current and updated novel food regulation, 2015-2283. Apart from traditional foods, which we mentioned before, we have 10 categories which span the whole range of possible new products appearing on the market, depending on the source they come from, on the process they derive from, but also whether they are, in the case of the last row, engineered nanomaterials, which was a novelty of the regulation, or specifically cell or tissue culture derived from living organisms, which also is a novelty of the updated regulation. During the consultation before this webinar, there were some questions from applicants on this novel food categorization. In particular, I think there was a request from whether precision fermentation would fall under. This to say that it is the applicant to decide during the application process to which of these categories they want to apply, one or more. Precision fermentation product we have been seeing so far fall mostly under the category produced from microorganisms, fungi or algae, but also new production process when they produce an ingredient which was traditionally produced in a different way. and also new or modified molecular structure. These are the macro categories pertaining to precision fermentation. And it is not unusual that a product may fall under different categories. We had the example of the first engineered nanomaterial authorized as a novel food, which was also a source of vitamins and minerals, and therefore appearing under two different categories. Still, the guidance provides for all the categories, for specific categories, all the details to be provided for the safety assessment of the product. The next slide, please. A bit of clarification on the authorization procedure, particularly for those of you that are not familiar with the EU framework. EFSA does not authorize novel food nor receive your application. The application are submitted for novel food according to the novel food regulation are submitted to the European Commission. It is the European Commission and the member state that decide whether the product is to be considered a novel food and not EFSA. The Commission can mandate EFSA to carry out the safety assessment of those products that consider to be novel. EFSA, together with its staff, its working group and its panel, dedicated panel, perform the safety assessment of this product with a legal deadline of 9 months, during which EFSA may request additional information to the applicant, and then EFSA issue a scientific opinion to the Commission and the member state for them to decide, take the appropriate risk management action and decide on the market authorization of the product and its integration in the Union list of authorized novel foods, which is finally the legal act where all the authorized novel food appear and are listed with their specification and proposed use levels. Next slides, just to give you a brief overview on what I was mentioning at the beginning. In particular, we don't come from this novel food guidance is not totally new. It's actually an update of the work we have been doing, particularly from 2018 onwards, when the current novel food regulations centralized within EFSA the assessment of novel foods. Before, this assessment was done by member states. Since January 2018, it is centralized with EFSA, and therefore we saw quite an increase of novel food under assessment by EFSA, driven mostly by decentralization, but also by the new provision to enhance, to protect, to give data protection to applicants, also protect the investment innovative companies make into filing an application to EFSA to enter the European market. You see the large share of the products we've been assessing recently in the last year are plant or their parts, but also products derived from microorganisms, fungi and algae, and products with new or intentionally modified molecular structures. We also got products derived from animal or their parts, particularly insects in the last years, and some recent cell culture or tissue culture derived from plant materials. The last part of, if we can go to the next slide, last part of my presentation will deal with some of the trends we've been observing in the area of novel foods, particularly to point at some of the areas to which we paid particular attention into the update of the guidance. For instance, novel protein and their source has definitely been a huge trend in our assessment. We'll see it later on, but also plant extracts are constantly in our list of product. Novel carbohydrates, I've seen a huge impulse, particularly derived because of precision fermentation also. But new processes are also there with less frequency, but they do appear in our dossier. And last but not least, nanomaterials or materials containing small particles or nanoparticles are more and more frequent also in our assessment. With regards to the novel proteins and their sources, so far these come either from traditional sources and processed with new processing, or from totally new sources from a food point of view. We've been assessing quite some insect species and several insects in general for different proposed uses. Several plant sources of new proteins, but also algae, microalgae and their biomass are very high in terms of dossier we are receiving. Byproducts and circular economy are certainly also another source of proteins we currently encounter. And mycoproteins or fungi, microfungi biomass is also an increasing trend that we are getting to assess in terms of novel foods. In terms of novel protein, you see they're listed the cell tissue culture, cell or tissue derived products. It is shaded because still the European Union has not seen any application of this kind of products from animal sources as a source of protein, basically. Other geographies do have, as you probably well know from media, but no application has been filed to the European Commission, and therefore no data have been submitted to EFSA. During the consultation phase before, sorry if you can go back, Ermo, to the previous slide. During the consultation phase for the product, we had some question also with regard specifically to novel protein sources. One in particular was whether a new protein isolate from commonly consumed food are to be defined as novel foods. As I said before, what is to be defined a novel food is defined by the European Commission and the member state, whether a food falls in this category, and therefore not by EFSA. What we have been seeing is that extract or concentrated forms of or some common food have been submitted as assessment for a novel food, because the Commission want to know whether the product, the concentration of the product or the product per se may entail new hazard. Recently we got, for example, potato protein concentrate to be assessed to see whether new hazard was inherent in the production process of this product. Next slide, please. Okay, novel carbohydrate is also quite a big trend we have been seeing recently. I put there two examples of macro categories. We do receive sugar replacer of different kind, oligosaccharides or single molecules. Most of these derive through enzymatic processing of other materials or through precision fermentation. This is definitely one of the highest contributors to the dossier we are getting, but we also observe a very specific category of products, human identical milk oligosaccharides intended for infant products. You see them listed there, those that have been assessed, and most of them already authorized by the European Commission. Again, a new class of carbohydrates which we haven't seen before in the area of novel foods. So definitely a trend from which we have learned a lot also for the update of the guidance. Next slide, please. We have paid in the guidance update particular attention to two macro areas which are surfacing as potential upcoming sources of novel food dossier. One being cell culture derived foods of animal or plant origin. As you know, this is already present in other geographies. In the EU, in Europe, UK and Switzerland got application, but not the European Union yet. Singapore was the first getting this an authorization for this product. US followed recently, but also other countries like Israel, for instance, gave already authorization. As I said, no application has been filed to Europe. As you know, this comes from advancement in the neighboring area of food, as it often happens for novel food. Cell culture derived food of animal origin come mostly from the combination of tissue engineering from biomedical area and cell culturing. It is an entirely new field of application in the area of food, but it does not necessarily entail new hazards, as far as we could see from the data that are available and the literature that is available. So we have developed quite some guidance for these products, spread over different sections of the guidance for new applicants when they will submit applications. And same applies for those products that are of plant origin, for which we got already 2 applications. The second area, which got quite some update in the guidance, derived from the impulse we got from precision fermentation product, microbial fermentation in general. Several of these products have been already submitted, deriving from microbial fermentation. Precision fermentation, as you know, does not have a regulatory definition, at least in Europe. In general, does not have an agreed definition, but it does refer to the engineering of microbial cell factories to produce specific food ingredients. In the case into Europe, the product derived from this methodology would fall under different regulatory framework depending on the intended uses. They may be GMOs when they contain genetically recombinant DNA. They would be food additives or flavorings, but when they are food or food ingredients, they fall under novel food. And therefore, you will see that quite a large update of the guidance it refers to this regulation. Sorry, to this class of products. If we can go to the next slides, this to say that we have been what you see here is one of the examples of the consultation we have run in the past months. The update of the guidance was not done in isolation. It did learn a lot from our past experience as a team, working group and expert within EFSA, but it does come also a lot from consultation with very different stakeholders. This is just one of the meeting we had. This was dedicated to cell culture derived food and food ingredients, where we met our stakeholders. You see, that was already May last year, to start to listen from them what were the specificity of their product, what were their needs in terms of guidance to develop product and file application into Europe. We did the same for other areas like insects. We did the same in other fora. Through the different engagement activities EFSA has put in place. And as I say, these were all steps to learn and get insight from the stakeholders on what they may be beneficial to be included in the guidance. Of course, we have listened a lot to scientists also, to the scientific community and updated science. And what you are participating into today is another event of this kind, which happened exactly in the middle of the next and the major engagement activities we do, which is the public consultation on what we have been doing. The guidance you have been seeing, preparing for this meeting has been largely updated, contains a lot of science and technologies development. We want to be also fit for purpose. Fit for purpose being exactly fitting the needs of those applicants that may apply, fitting the need of risk managers that will need to use the guidance. And this, I think, is also one of our core mandate with the revision of this guidance, which we aim to achieve also with this public consultation. So I thank you for your participation again, and I now pass the floor to Hermalos for the next part of the presentation.
Thank you a lot, Andrea. Good afternoon to everyone. So now we're going to talk about the novel food guidance update. What is the purpose of the update, and to discuss together the overview of the proposed changes. In the past few months, we have been working here at EFSA, I could say quite intensively, together with the experts of the Novel Food Working Group and also of the NDA panel, to prepare an updated proposal for the new Novel Food Guidance document. As Andrea told you before, the initial guidance was issued back in 2016, and since then we have updated only once in order to accommodate the needs of the transparency regulation back in 2021. So all this work is done to address a mandate we have received a few months ago, last summer, by the European Commission. And the main reasons for requesting this update were some changes regarding the regulatory framework of novel foods, some implementing regulations. Of course, the Commission asked EFSA to embed its experience in assessing novel foods in this new novel food guidance document, and also to consider advances in the field of food science and technology. And regarding the EFSA experience, since 2018, there is a centralized assessment of novel foods in the European Union done by EFSA. So this means automatically that we have assessed several heterogeneous novel food application dossiers. And also, since then, there are many new EFSA cross-cutting guidance documents applicable, which also which are relevant for the novel food risk assessment. And also, in general, there are methodological advances in the field of risk assessment. Also, EFSA has developed new tools that can contribute to the risk assessment of these regulated products, for example, in the area of exposure assessment. And also, as Andrea told before, we have been receiving constantly feedback from our stakeholders through different means. For example, the EFSA scientific colloquium we had last year, but also through the clarification request teleconference that we have with applicants during the risk assessment through Ask EFSA. So we try to embed all this knowledge, all this information in this new updated guidance document. And it would be nice to say that in parallel, we have also another separate administrative guidance for the novel food dossier submission, and it's also going to be updated together with this scientific guidance, and it's another unit of EFSA, the FDP unit, who is taking care of this update. And in this guidance document, the topics tackled at some administrative aspects relevant to the transparency regulation requirements, and also to some aspects like the e-submission tool and things that the applicants have to take care from an administrative point of view. So now, briefly, the timeline, we are towards, let's say, the end of this updating process. Today we have the stakeholders event, this webinar, and I hope that most of you have already gone through the guidance, the draft guidance that was endorsed by the NDA panel end of January, and it's out for public consultation since mid-February. That is going to last for 2 months until mid-April 2024, and the plan is to have the guidance adopted by the NDA panel in June of 2024. So now I'm going to move through the different sections of the guidance and give feedback to you with regard to the major updates done. So in the area of identity, we have tried to expand and explain more some key aspects, key concepts linked to the identity. So first of all, we have tried to provide guidance on what should be considered as the novel food subject to the risk assessment. And in this framework, we have tried to explain in which cases non-novel ingredients, for example, mixed with the novel part of the product, are considered part of the novel food or not. And also we have tried to provide some insights for the applicants on how to name their novel foods, so to have a more accurate description of the different products, and also to avoid claims in the nomenclature of these novel foods that are subject of other regulatory frameworks. On the screen now you can see the 6 macro identity categories. We have created a category putting together chemical substances, novel foods of mineral origin, and polymers. There is another section for novel foods consisting of isolated from or produced from microorganisms. And we have created a section for novel foods produced from plants, macroscopic fungi, and macroscopic algae or their parts. Before they were clustered in different way. There is also another identity section about the novel foods from animals or their parts. And as it was mentioned in the previous presentation, we have provided in this draft proposal additional information on establishing, investigating the identity of cell culture or tissue culture derived novel foods, either from animals or from plants, macroscopic fungi, and macroscopic algae. And this section actually has two subsections in the identity with more specific requirements for those two clusters. And last but not least, we have the identity section about the novel foods that contain or consist of engineered nanomaterials. Moving to the slides with some more details. Regarding the first cluster, chemical substances, polymers, and novel foods of mineral origin, we have added some insight on the provision of new identifiers for this cluster of products. Also provide some insight about identification methods and techniques to be employed for investigating the identity of the novel food. Also, some information about the suitable comparators when trying to prove the identity of these novel foods. And also, we make reference to the ECHA, European Chemical Agency guidance on identifying and naming these novel foods, these substances. In the section of novel foods linked to microorganisms, we have tried to explain the different roles that microorganisms may have in the novel food production. Regarding novel foods produced using GM production strains, we need more information on the purpose, characterization, and structure of the genetic modifications. And also there are some additional testing requirements regarding the presence of viable cells and DNA of the production strains. Of course, we make reference to the QPS status, the qualified presumption of safety, and also to the requirements, identity-related requirements about whole genome sequencing for these products. Then, regarding the subsection of identity for plants, macroscopic algae, and macroscopic fungi, we give some information on how the identity can be experimentally verified and how the growing regions and the harvesting season may impact the identity of the novel food. Also, a new element introduced here is the need that we have a non-GMO statement for this type of novel foods. And actually, you know that in the European Union, GMOs are different by law from novel foods. However, this statement could also help in the validation stage of the dossier at the European Commission level. Now, moving to the other three subsections of identity regarding novel foods produced from animals or their parts. Again, verification of identity, information on the suitability for human consumption, as well as information on the animal's health status. Also, we need to know the origin of the initial livestock, and same as before, there is a request for the non-GMO statement. Regarding the cell culture or tissue culture dry foods, we have created, as we said before, two subsections, and there are requirements, identity requirements for the cases where established cell lines are used or primary cells are used. Also, we request evidence for compliance with inspection requirements and absence of zoonotic agents. And also, we request information on whether the cells or tissue sourced from the non-GM source have been genetically modified. And in the section, identity section of engineered nanomaterials, we make reference to the new EFSA guidance on risk assessment of nanomaterials to be applied in the food and feed chain of 2021. And also throughout the whole guidance, there is a there are insights on distinguishing between small particles, including nanoparticles, and engineered nanomaterials. Moving now to the production process, we have, let's say, 4 subsections, one with general provisions about the input materials, production yield, novel aspects of the process, quality and safety assurance elements, as well as standardization criteria for producing the novel food. There is a subsection about considerations for specific production process steps, where we ask the applicants to describe conditions of farming practices, culture conditions, biological agents linked to the novel food production, post-harvest handling procedures, inactivation or removal of food enzymes, as well as there are some specific requirements regarding the status of the regulatory status of food enzymes used in the production process. In the section about consideration for specific novel food categories, we have provided some insights regarding the production of different novel food belonging to different clusters. For example, different consideration when you have something that is chemically produced, is chemically synthesized, or something that employs in the production process microorganisms. And last, in the subsection additional considerations, we give some insights to the applicants, of which the application includes several producers for the same novel food, and also some information on what happens when the applicant does changes in the production process during the risk assessment, and also what an applicant should do if they change the production process after an eventual EU market authorization. Now moving to the section of compositional data, we have tried to explain what is the role of this data in the risk assessment, and we will discuss this in details in the Q&A section later. And you can see here we have different subsections, general requirements, where we talk about the analytical methods to be used, how to address compositional variability among batches, which are the best sampling practices, and also information on the compositional analytes, which parameters the applicants need to analyze to address the identity safety of their products. We have kept the previous section on single substances, simple mixtures with some modifications, as well as on complex mixture and whole foods. And last but not least, the stability of the novel food. We have introduced some elements according to our experience so far. And also we have created a new subsection on the impact of processing on the novel food when it's used as an ingredient in various proposed for use matrices. Now, regarding specifications, we have tried to explain the role of the specifications for the novel food risk assessment. Specifications encompass chemical, physicochemical, nutritional, as well as microbiological parameters that define both the identity of the novel food as well as the safety of these products. And we have provided some information on the role of specifications in risk management, and so that the applicants can do more informed choices when proposing both the specification parameters as well as the respective limits, the respective levels. Then, regarding the history of use of the novel food and its source, 2 different, let's say, macro areas. The history of use of the novel food. We need to know how the novel food is used as food outside of the European Union, if there are any non-food uses, the extent of its use, the population groups for which it has been a dietary component, its role, of course, in the diet, and as well as information on handling and preparation methods. And regarding the history of use of the source, we need information on the composition of the source, on different production processes that are linked to the source, and also if available experience from the use of products that derive from the same source as the novel food. Now I'm moving to the section of the proposed uses and use levels and anticipated take of the novel food. Here actually we have some amendments due to the implementing regulation of 2017. Now the target population of the novel food, when the product is added as ingredient in other foods, or when the novel food is going to be consumed as a whole food, cannot be any more restricted. So the novel food has to be safe for the whole population Of course, this does not apply in cases where, for example, the novel food is going to be used as ingredient in food supplements. We have also the section on the proposed uses and use levels, where we present some requirements specifically for food ingredients, whole foods, food supplements, and also particular food categories. And also in the displayed take of the novel food, we have embedded information on new tools that can help to perform the exposure assessment. And also we have modified the section of combined intake considering other sources of the novel food or its paid constituents, as well as the section regarding the estimated exposure to undesirable substances. Now, in the next slide, you can see the complex, let's say, tiered approach for ADME, absorption, distribution, metabolism, excretion, and toxicity testing. Actually, it may seem complex, but we have tried to give clear instructions on the different macro areas, ADME, genotoxicity, and repeat dose toxicity, as well as you can see that there are 3 different tiers per area. So there are different triggers that request to pass from tier 1, for example, to tier 2. And you can see that sometimes there are triggers linked to additional testing, for example, from ADME to genotoxicity. So there are different parameters to be considered, but we will see more in more details the information on those 2 macro areas in the Q&A section. And of course, we have amended the subsection on human studies, providing guidance on both how to use existing evidence from human studies, as well as for conducting new studies to address the safety of a product. Now I'm moving to nutritional information. Actually, one of the main things we have done here is to explain what means nutritionally disadvantageous, to give more clarity to this concept in the novel food risk assessment. So in the whole section, you can see here there are different subsections regarding the excess intake of nutrients, inadequate intake of essential nutrients, specific considerations for novel foods, proposed and new sources of micronutrients, specific considerations for novel protein sources, as well as some additional information. There are different requirements in each subsection, but what we try to implement also in this section is a pragmatic approach that is feasible for the applicants. It's also meaningful for us doing the risk assessment, but it's also useful and informative for the risk managers and the policymakers later. And some of the elements, we will see them afterwards, again in the Q&A, according to the question you have submitted. But I would like to show you this flowchart. We provide through this flowchart guidance to the applicants on how to investigate the nutritional impact of the novel food, of course, always under the proposed uses and use levels. And this is the last slide about the update overview. The allergenicity section. This section has 2 objectives. First of all, to inform risk managers about allergenic properties of a novel food, which may serve them for their marketing authorization later decisions, including those with regards to possible labeling requirements. The second purpose of this section is to collect all the valuable evidence related to the allergenicity of a novel food, and also to generate a limited set of data in order to gain some knowledge about the respective allergenic properties of the novel food. And we have divided this section in 4 subcategories based on, let's say, the protein situation for each product. So we have section 1, novel foods with no protein content derived from the production process, novel foods from allergenic foods subject to mandatory allergen labeling with no proteins from other sources, novel foods derived from allergenic foods which are not subject to mandatory allergen labeling, and then the last section is about novel foods for which the allergenic potential is unknown. And there we address single protein and simple protein mixtures, and also complex protein mixtures and whole foods. And of course, we can anticipate that for each one of these different subcategories, there are different data requirements. So this is actually the last slide, as I told you before, for the update of the guidance, the overview of the main changes. So before moving to the next section, the Q&A session, when we're going to reply to your questions. We will have a short 10-minute break. Please do not disconnect from the meeting, and be back at 5 to 4 to continue with our webinar. Thank you a lot.
Welcome back. I hope you had the time to stretch your legs. So this is the main part of our webinar. And I hope the initial presentations gave you the background and a bit of information of the main changes that we are trying to implement in this draft guidance. Now, we're really going to go through the questions and answers so trying to answer to the questions we have received. We have, in fact, received quite a substantial number of questions. I hope there is a nice slide so I can show the numbers. All right. So we have received 181 questions. They were submitted by 76 of our registrants. We try to classify those questions in categories very much as the guidance document has been organized. So we have a few questions on production process, some on identity, and so forth. There are a number of questions which we consider they are out of the scope of this event, mostly related with risk management questions which need to be addressed to our colleagues in the European Commission. What I would like to say is that, as I mentioned already, please address your questions also, your comments or your suggestions also during the public consultation. This would be most welcome and very much needed. However, we'll try and address as many of the questions as possible. As you can imagine, we cannot go through all these questions, but we try to cluster, and I hope you find this useful. And without any further ado, I will ask my colleague Andrea Germini to take the first lot of the questions. Andrea, the floor is yours.
Thank you, Anna. Let me just reiterate what Anna was saying. We have been trying to answer by clustering all the questions we received. If you don't get an answer or a clear answer from this webinar, please post your question into the public consultation tool, because that will be essential for us to take care because it does mean we have not conveyed the proper message, neither in the guidance nor here. So I will start with identity, and then I will follow up with the production process section. We clustered the comment around the bullet point you see here, so I will touch upon them one by one, trying to address the combination of the question we got. So the first one has to do with the status of products that are produced with multiple sources, being novel or not novel. In particular, the applicant was asking whether different plants could be part of the same novel food. Yes, that is the case. It is a mixture of different sources is indeed possible. We have received different combination of novel and non-novel sources and already assessed quite some of them. Some cases recently we had pea and rice protein extracted from the two different vegetables, but also combination of different root extracts from exotic plants. The second point has to do with the role of non-novel components in the product to be assessed. As you have heard before, and is clarified in the guidance, we have specified clearly what is the novel food. The final product may contain non-novel components, which are also put together with the novel food. And therefore, we are mostly interested, the applicant was asking, should interaction occur with the non-novel part of the product with the novel part? How it should be considered in the evaluation. I think, first of all, the applicant should explain the purpose of these substances and clarify whether these indeed react with the novel food and what are the implications for the safety assessment. This information is needed for us and the working group to discuss then the implication this might have on the final product. Origin of the novel food was also a question, whether EU or global. origin may be whatever part of the world. So it is not restricted to EU. The reason why we do ask for origin, specific origin, is that origin may provide information on the possible presence of specific environmental contaminants or specific soil components, which may trigger specific alert during the assessment and identification of potential hazards. The next bullet point has to do with the use of whole genome sequencing in identity and when this is required for novel food assessment. In the guidance, the whole genome sequencing is required. It is mandatory when the microorganism is the novel food under assessment. We had few cases for that. But also when the microorganism is used to produce the novel food, so is a producing mean, or it is, or the production process of the novel food uses an enzyme not yet assessed by EFSA, produced through microbial sources. In all these cases, whole genome sequences is required for the novel food assessment. Next question has to do with the purity levels that are accepted. This really depends on the novel food. I mean, it's really on a case by case. For instance, if we take a microorganism, the purity level has to be defined as strain level. The level of identity has to go down to the strain level. For complex botanical mixtures, these may depend on the fraction of the uncharacterized material. The larger the fraction, the highest of uncharacterized material, the highest the purity required is. So it is defined in the guidance. You'll see clarification of this, but it really depends on a case by case on the type of novel food you are asking assessment for. Next is taxonomy, and in particular the question, if I'm not mistaken, was about algal biomasses and the products thereof obtained from these products. It can be extended in general. Particularly algal and microalgae have some nomenclature issues because some of them are not fully defined. In general, the guidance provides indication for a reference database for all the possible sources of novel food. So these are at the moment the most known resources that you can use. They are available there. In the future there might be others, but the one we are using as a reference are listed there. When the classification is still controversial, as it is the case for some microalgae or algae, then the applicant has to support its argument for the taxonomy identification with scientific evidence. And our experts will assess this together with the rest of the information and then try to make sense of the taxonomy of the product. Another question then was on the use of GM microorganisms in the production of novel foods. And particularly, the question, let me read it, was on whether an already authorized novel food is replaced by, usually produced with a non-GM microorganism, is now produced with a GM microorganism, but the food product obtained is identical to the one already authorized. What the applicant has to do is to demonstrate first the identity of the final product, meaning the product obtained is exactly the same as the one authorized. And second, clear the safety of the product by assessing the safety of the producing strain, the new producing strain, ensuring at the same time absence of viable microorganism and absence of recombinant DNA in the final product. The guidance also has introduced a demonstration of non-GM status for some categories, particularly those for which cells can be taken for further growing. And the question was how to demonstrate this non-GMO status, and is an external validation necessary? External validation is not, strictly speaking, necessary. A statement by the applicant may be enough if accompanied by the information on the source material and experimental evidence provided by the applicant using validated methods. This would be enough, and external validation of non-GM status is not necessary in this case. Last part of the identity has to do with engineered nanomaterials and nanoparticles, if you wish. In particular, the requirement for what it is needed to provide information that the product does not meet the definition of engineered nanomaterial. The guidance in this case refers to the EFSA guidance on the safety of nanomaterials for all those products that meet the definition of the European Commission. So if the definition of the European Commission, which has clear technical requirements, is met, that guidance, the guidance on the safety of engineered nanomaterial, has to be used. For products that do not meet the definition of nanomaterial as provided by the Commission, by the European Commission, then potential presence of small particles and nanoparticles is assessed through a tier approach, which is described in a dedicated guidance, different from the engineered nanomaterial one, which starts with a simple solubility test. So it's a tier approach starting with a solubility test that the applicant has to provide. This is particularly applicable to powder material in general, all this area of nanomaterial and nanoparticle more in general. If we can go to the next slide then. And the production process. So basically here the registrants, one of the common question we got was the required level of details in the production process description. That is a tricky issue we got also from the stakeholder meeting we had. So first of all, let me remember all of you that the production process under the Novel Food Regulation can be covered by confidentiality. So the opinion discloses only a summary of the production process information, which is in general agreed with the applicant before final publication. This to say that in order for our expert to evaluate the impact of the production process on the safety of the novel food, we need all the details of the manufacturing process. Again, this is covered by full confidentiality if you require so, and therefore nothing of what you send to us get disclosed to the public. But we need all the details in order to identify any potential hazard inherent to the production process. Some requested information on HACCP and food safety management system in general and their role in the assessment. In particular, one of the requesters was asking whether a certification issued by an accredited organization, certification for food safety management system such as HACCP would be enough. And the straight answer is no. I mean, we do ask for this information, but we also want to see the plan, the monitoring plan, in order to assess the presence of potential hazards. And therefore, we need the details of the control plan at least, and the critical control points and monitor parameters. This is one of the things we look at when we identify potential hazards in the production process. There was a question on a recent addition we have in the guidance on the compliance with food contact material regulation. I remind you, it has regulation 1935 2004-26. In fact, every material that goes in contact with the novel food has to meet the requirement of this regulation. And this is because we have noticed through the assessment of some products the presence of contaminants, for instance, in the final product, which we would have not been expected given the production process, nor given the nature of the source of the material. And by investigating further, we found out that this was due to contact material being in contact with food not being certified as a food contact material. So food contact material use are mandatory for the production of food, and therefore we need a certification, a statement on the compliance that the material you use are indeed certified to be in contact with food material. There was a question on the information on raw material, in particular, what are the expectations with regards to the amount of information required? And the straight answer is the information has to be as detailed as possible. It is to ensure that the experts have, in general, I mean, all the information to assess potential presence of hazards. This does not apply only to raw material, but all the input materials that get that are used in the production process. So, for instance, the purity of a solvent used in an extraction process also has to be provided, same as a solvent used in organic synthesis, for instance. The purity of those solvents is to be provided, considering this as input material. Same applies to all material that enter into the production process. There were some question on the use of food enzymes in the risk assessment. Use of food enzymes in the production of the novel food and the way they are risk assessed. And particularly, the questions, there were a couple at least, were referring to enzymes not yet assessed for their safety by EFSA, or enzymes obtained by microorganisms that are immobilized, so specific examples. Or another question of food enzyme was whether there was an harmonized list of approved enzyme into the EU. So let me start with this last one, and then I'll go through the rest. Unfortunately, there is not yet an established list of authorized enzyme at EU level. This should come in the near future, is under preparation by some times. Still, there has been already quite a big amount of work done in the assessment of enzymes in some countries and also in EFSA. So in the case of novel food, whenever the enzyme has been assessed already by EFSA, by the panel by the SEP panel by now, that assess the safety of this enzyme, we take this information into consideration. If this is not assessed by EFSA already, we do treat the food enzyme as an inherent component of the final novel food. We do characterize the novel food in an holistic manner, and we request information also on the food enzyme itself and the microorganisms used to generate this enzyme. All the details are in the guidance document of EFSA. Linked to this, some of the requests are also requested why the EFSA feed-up panel is mentioned in this area of food enzyme and microorganism assessment. This is referred to in our current guidance because to date, feed-up panel 2018 is the most up-to-date guidance for the assessment of microorganisms, and therefore all the regulated areas in EFSA still refer to this guidance. For your information, in fact, EFSA has already started a big project of revising all the microorganism assessment, taking stock all the requirements of the different regulated product area and beyond that. And hopefully by the end of this year, a overall EFSA guidance for microorganism risk assessment will become available, which will cover the needs of all regulated areas. And this will also cover novel food when this will be available. For this, at the current present time, EFSA feeder panel 2018 is still the most updated, and it is the one you should refer to while waiting for the new guidance. The second last bullet point has to do with the production process scale. This is a recurring issue we have encountered also in the meeting we had with our applicants, with our stakeholders in preparation for the guidance. It is extremely relevant for microbial fermentation, precision fermentation, as we can call it, tissue culturing also, because most of these products are currently not yet at industrial scale, but it is in general a problem also for other categories of food, because innovation starts at pilot scale, research and development scale, and therefore we recognize the need, and the Commission recognizes the need also to take this into account. Let me also state that the novel food guidance, the novel food regulation stated the assessment of the safety of the product has to be done on the product as intended to be placed on the market. Therefore, in theory, that should be the industrial scale or something as close as the industrial scale. We know if you lack market authorization, particularly small companies, it's highly unlikely that you are at the industrialized scale, but they are at the pre-industrialization stage. So in the guidance and in the recent past, we have accepted also batch analysis and production process that are at not yet at industrial scale, at pre-industrialization scale, when this was clearly linked with information on the intention to scale up at industrial scale the pilot, and what changes would the production process have gone through while scaling this up. This also goes with the question we got whether it would be possible to validate a production process mock-up, as it was called by the applicant, by the requester. Strictly speaking, we don't validate production process mock-up, and we cannot validate a production process that is not there yet when it is industrial. But we do recognize the value of pre-industrial batches. We use those for the assessment together with all the information you provide for possible scaling up. Still remember that there is into the novel food regulation a precise article, Article 25, which requires the food business operator to notify the Commission of any changes of the product placed on the market from a safety and technical point of view that may influence the evaluation of the safety of the novel food. So ultimately, it is the food business operator responsibility, while complying with the specifications set for the product eventually authorized with the pre-industrialization stage process, to comply with the specification or notify the Commission if they believe that there are changes introduced in the process while scaling up that may affect the safety. And then the Commission may decide with member states whether the changes may deserve indeed an assessment, further assessment of EFSA, or can be considered as not impacting on the safety. This links well with the final bullet point I have here for discussion, which are the changes in the production process during risk assessment and after the risk assessment. We had encountered these cases also during risk assessment. If these happen during risk assessment, EFSA should be immediately provided with the details of the changes and the new production process, the full details, together with the compositional analysis of the resulting batches, to see the impact the changes in the production process might have brought to the safety of the product. Remember, these may, as a second line, have an impact on the safety test, because the test items used for the safety has to be compliant with the specification and the requirements that are set for the product. So if you may need to perform the novel safety test if the test item has changed substantially composition, or if new hazards have been introduced. If instead this happens after authorization by the Commission, then the Commission should be immediately notified. And this again is linked with the Article 25 I mentioned before, meaning it is your food business operator responsibility to notify of any change and the nature of the changes. And then the Commission may decide, or may decide not to, may decide to send, to request EFSA to evaluate the impact on the final product. We know there is quite a demand, particularly with the stakeholder we have recently met in the area of microbial production, fermentative product, but also in other areas, to constantly optimize the process, change cell lines, change media components, change production strains. Again, the regulation requires us to assess the product as they are intended to be placed on the market. Therefore, we have there is little margin for us. There was a request linked to this that there could be a light or a quick notification or evaluation procedure when only minor changes are implemented, such as a new strain or a new media component. Such procedure is not in place in the current regulation and does not depend on EFSA. But we have collected similar requests during our consultation. We are in constant consultation with the Commission, so we have reported these needs, and there might be future actions from the Commission, but these do not depend on EFSA nor on the guidance we are discussing. For your information, the current regulation already foresees the procedure to change specification, change production process, for example, the use of a different strain, or change modification or propose modification to the use that are in the union list. But all these processes passes through the Commission, and there are clear criteria to what then is required to assess the safety of the new product resulting from these changes. I think with this I cover all the points that were sent in these two areas. And therefore, I will pass now the floor to my colleague Hermolaus, which will follow up with the compositional analysis. Hermolaus, floor is yours.
Thank you a lot, Andrea. So now we're going to tackle the topics raised by the registrars regarding the compositional data and the specifications of the novel food. One of the questions raised was, what is exactly the role of compositional data in the novel food risk assessment? So many times we say that the compositional data is the backbone of a novel food dossier, because they have, let's say, multiple functions in the novel food risk assessment. First of all, the compositional data help to characterize the novel food and its constituents. And they are there to address the chemical, physicochemical, microbiological, as well as nutritional composition of the novel food. And the compositional data should allow for an in-depth exploration of what is the content of this product. And we use the compositional data to identify possible hazards, to set up, to fine-tune the specifications proposed by the applicants, but also the applicants can use the compositional profile of their novel food in order to, let's say, decide on what will be their ADME strategy, their toxicological testing strategy and the approach that they will follow, as well as to have better insights in regarding the nutritional information of the novel food. Then in the second point, actually it was something that was requested by many registrants, was to clarify better the number of batches to be analyzed by the applicants. So in the previous guidance, it was written preferably at least 5 batches to be analyzed. And now the experts of the working group of novel foods and also the NDI panel, they say that we need at least 5 batches of the novel food to be analyzed. And apart from the number, some people also requested clarifications on the word independently produced, especially in the case that the production process is a continuous process. So regarding the number, it's linked to the fact that we have to address, actually we have to assess, but the applicant has to address eventual compositional variability of the novel food among the different batches analyzed, and also to both the applicant but also for the risk assessors to control what is the repeatability, the consistency of the production process. If the producer is able to produce the novel food with constant desired compositional characteristics. And also through the 5 batches, it will be easier to investigate any, let's say, the representativeness of the compositions regarding the final product, and also having at least 5 batches of the novel foods analyzed. It will make it easier both for the applicants but also for the risk assessors to detect any possible outliers that come out through the compositional analysis. And actually, in the past, we have seen examples of novel food dossiers containing less than 5 batches. Some of them they had compositional parameters with levels very far from the other batches, so it was very difficult to tackle these differences, to investigate them better, and also to see how meaningful they are. So it happened often that we had to go back to the applicant to request for analysis of more batches. And regarding the independently produced batches, we mean that every time there is, let's say, a new production cycle, new raw materials are used to produce the final product. But of course, in the case that the process is continuous, it should be the applicant to, let's say, to identify some key parameters to, let's say, tackle this point. For example, it could be some key points of the production process. It could be something that is linked to different time intervals of the production process, or it can be something linked to, for example, a predefined quantity of the novel food produced. But also in this case, the applicant has to provide to EFSA the reasoning for the different sampling and the nature of those batches analyzed. Then there was another question about using omics in compositional characterization of the novel food. Actually, regarding analytical methods, it's written in the draft guidance proposal that EFSA prefers that the novel foods are analyzed using validated methods, preferably recognized at national or at international level. Of course, we acknowledge that here we deal with novelty, so maybe there is not an official method to analyze something, or maybe there is a method to analyze something but is validated for a different matrix. For example, a method to analyze fat in milk can be applied, for example, in a novel, let's say, in a novel food or in liquid form. But in this case, we need the applicant to provide to us elements on how this adaptation of the method has been validated. And in case that, for example, the applicants want to use some omics approach, for example, to do some untargeted analysis, in case that such methods are not validated, they can be still used to, let's say, to investigate a bit the profile of the novel food. So the data produced by omics methods, if these methods are not validated, can be used, let's say, as supportive element for the risk assessors and also as material for the applicant to start investigating better the compositional nature of their products. Then there was a question about the cases where there are components in the novel food production process which have not been previously used for food production. And in this case, we will need to have detailed information on these components, for example, the specifications of these raw materials, and also to know how much of these components result in the final produced novel food. And then, of course, depending on the findings, there may be additional requirements for ADME or toxicological or other types of testing. I'm moving now to the next bullet point about addressing compositional variability among the batches. We have encountered cases that the composition varies according to the season, for example, season of harvesting for plants. We have variability due to differences among the raw materials used in the production process. And in some cases, even operational parameters of the production process may slightly change, for example, a range of temperatures, and this could lead to variability among all batches analyzed. So the applicants, when they are doing the sampling of their batches, they have also to consider this element, that they need to address the compositional variability among the batches in order to have, let's say, a more representative pool of analytical data for the composition of the novel food. In the next bullet point, another element requested clarification was how thorough should be the compositional characterization. How much should I analyze? How many analyses should I do? So actually here, it's somehow linked to what Andrea told you before, how detailed should be the production process. So let's say the more, the better. Of course, this can be said on a case-by-case basis by the identity of the novel food. For example, we expect differences when we deal with a single substance and differences when we deal with whole foods, because we know that whole foods cannot be readily characterized like 100%. It's impossible. But in this case, the more data we have on the composition of the novel food, the better, because actually this body of compositional data, compositional evidence could also, let's say, trigger additional testing, for example, toxicity testing, or in some cases, a very thorough compositional characterization can lift the need for additional toxicological testing. Then there was another question, another topic about the sources for hazard identification, like what the applicants can use to have an idea of what are the hazards that could be present in their product. So in the guidance we are making reference to some, let's say, databases, to some sources, for example, the Compendium of Botanicals, some other databases. But of course, the scientific literature is a very big pool from which somebody can find information on possible hazards present in their product. So in this case, let's say it's up to the applicant to do a very thorough search to identify, let's say, this kind of type of hazards. Of course, EFSA has some platforms, for example, the Open Food Tox database, where you can find some information, as we said before, the Compendium of Botanicals, but it's also very, very important to look at previous EFSA scientific outputs. For example, if there is a novel food opinion on an insect-derived protein concentrate, and then another applicant wishes to apply for a similar product from another insect, it would be very useful that they look at the previous output to see where EFSA and the experts have put emphasis when performing the risk assessment. The next bullet point about the analytical methods accepted is somehow linked to what we said before about omics. So preferably validated methods or nationally or internationally recognized. Of course, we need to know the limit of detection of the method, the limit of quantification of the method. And in cases that an applicant uses in-house methods, for example, a method developed for a specific component of their product, then they have to provide the full description of the analytical protocol, the full description of the method, all the steps followed, as well as data that prove that this method has been validated. And of course, this type of information will be assessed by the experts of the working with the panel. And in case that something is needed, then we will go back to the applicant. Then there was another bullet point about appropriate comparators for in the area of compositional information. And actually, the word comparators appears also in different areas of the novel food guidance. But in the case of the composition, it really depends on the novel food to be assessed. For example, we can have comparators, appropriate comparators based on the type of product, based on the uses of the proposed use of the product, but also in some cases we can define, the applicant can define appropriate comparators, for example, when looking for existing legal limits in the, for example, in the European regulatory area. For example, if the novel food is an oil, they can find some comparators would be oils already authorized, already used in the EU, to at least get a grasp of permitted levels for substances or other types of regulatory limits. And then based on this information, they can also decide to launch additional analysis for their product. The last two bullet points have to do with the specifications of the novel food. And actually it was requested how specifications are linked to the compositional data. And actually, first of all, the compositional data, the third compositional data serve to establish the specification parameters. And these parameters have to be, let's say, established based on the levels that somebody is analyzing. For example, if the protein level content is between 10 and 15% in the analyzed novel food, then it would be very irrational to have a specification limit of 10 to 90%. So the compositional data have to be considered when establishing the relevant specification parameters, but also their respective limits or ranges. And of course, for example, when an applicant does the stability testing to see how the product behaves during the storage time, during the proposed shelf life, then the applicant should also have in mind that also during the proposed storage time, the proposed shelf life, the product has also to comply with the specification parameters. So it's not only the batch-to-batch analysis of the produced product to be considered, but also the analytical data occurring from the stability testing of the novel food. And there was another question, how is tackled the fact that the specifications are proposed on a specific, using data for a specific product, but then the authorizations are generic. So actually this is something, let's say, between risk assessment and risk management. So at EFSA's risk assessors, we see which are the proposed specification limits and parameters by the applicant, and then our experts, if it's needed, they propose amendment of specifications. So what will be seen on the final output of the risk assessment will be what the working group and the panel propose to have as specification parameters and limits. And we should not forget that here at EFSA, we risk assess a specific product produced using a specific production process. Of course, if there are some key parameters linked to the production process to this product, they should be introduced in the specifications. When the output is ready, then the risk managers, they take the proposal from EFSA and they can decide to keep it as it is, to put additional parameters, to exclude some parameters, to keep the levels EFSA proposed, to expand or to reduce the levels. So then it's up to the risk managers to take something specific and try to adapt it to something more generic. So these were the information about compositional data and specification. And now I will give the floor to my colleague Emanuela to guide you through the history of use, proposed uses, use levels, and anticipated intake.
Good afternoon. I'm pleased to guide you through this Q&A part on history of use and exposure. Let's start with the history of use of the novel food. How can I document the history of use of my novel food? This was one of the questions we have received. Via publications, you can provide publications, meaning articles, books, which indicate in which countries outside Europe your novel food has been consumed, since when your novel food has been consumed, and how its role in the diet, the handling, the preparation. This type of information should also be provided for the source of your novel food when it's applicable. Another type of documentation that you should provide are authorizations, for example, that you have received outside Europe on your novel food. I just cite as an example, if you have applied to USA for a GRAS status, you should indicate this in this section. You should provide the notification, the GRAS notification you have submitted to FDA. I know that in the administrative part, you are also requested to provide a regulatory status of your novel foods, but also here it's relevant if you make reference to the authorizations you got on your novel food outside Europe. You also need to comment whether the uses of your novel food outside Europe are similar or they are different from the intended uses here in Europe. Another point, if in the country outside Europe there is a surveillance system in place, you should also provide information on potential alerts you have received on your novel food outside Europe. So now let's move to the exposure part. There was a question asking about tools available to anticipate the intake of the novel food. Yes, they are available. They are mentioned in the guidance. We have the FAME tool, a tool which has been developed by EFSA already some time ago for food additives. It's an old acquaintance because it's already mentioned in the current guidance dated 2016. We refer again to this tool here in the guidance, in this update of the guidance. Since we share, let's say, common features, we are also interested in novel foods on chronic exposure of foods. You can use the FAME tool also for novel foods. Then there is a new entry. A couple of years ago, EFSA developed and made available the Dietex tool. It's a tool which is publicly available, and it offers you a greater variety of foods as compared to the FAME tool, which being developed for food additives, it only, let's say, restrict the uses to the food additives regulation. So these are the two tools that you can use to anticipate the intake of your novel food in Europe. Second bullet point, exposure assessment. Here there was a question asking, but which type of data do I have to provide you regarding the intended uses and the exposure? First of all, you need to clearly indicate which are the intended uses of your novel food. For example, if you want to place on the EU market your novel food as a food supplement, you need to indicate this. You need to indicate, to specify the maximum daily intake of your novel food and also the target population. If you want to add your novel food as a food ingredient in foods, Here you need to indicate, to list all the food categories in which you intend to add your novel food, plus the maximum amount of the novel food that you want to add in each of these food categories. So depending on whether you are using the frame tool or the Diatex tool, you need to indicate this list of your intended uses. Then, I mean, these two tools which are available, they once you impute this data, you select the food categories and you impute the maximum amount of the novel food, the tools generate an Excel table with the exposure, with the anticipated intake of your novel food in Europe. Because behind these tools, there are consumption data collected by EFSA through European national dietary surveys. So at the end, you really end up with an Excel table reporting the average and the 95th percentile of intake of your novel food on a milligram per day or in a milligram per kilogram body weight exposure. And you need to provide in your dossier this information, including also the Excel output that you got from the FAME tool or the Diatex, depending on what you are using. Then behind this bullet point, there was another question asking whether EFSA would complement the exposure internally. No, I mean, once you give us, you tell us which are the intended uses. If you intend to add your novel food in foods, and you tell us the list of foods in which you want to add the novel food, the maximum use level, we repeat the calculation, the estimate via FAME tool or Diatex tool, depending on what you have chosen. And we check the output. If there are inconsistencies, we come back to you. Then we share the output of the exposure with the experts in case they may have questions. And if they have questions, we come back to you in a clock stop letter. Then another question behind this bullet point was whether human testing was necessary for the exposure. I mean, it's wrong probably to call it exposure. Here we are dealing with the anticipated daily intake of the novel food. So for this purpose, we need to estimate the intake of the novel food once it will be placed on the EU market based on your intended uses. So here we really we recommend you to use these two tools. However, human studies are really necessary for a novel food in a novel food application. In fact, there is a dedicated section in the guidance where you are requested to provide human data on your novel food. Then let's move to the third bullet point, authorized uses, novel foods versus food additives. Here we received a question asking whether at the time of the authorization, the requester was highlighting that it would be helpful if at the time of the authorization, the entries in the union list would match the food categories of the food additives regulation. I just want to make a remark because first of all, I mean, the authorization phase of a novel food is outside the remit of EFSA. However, I just want to make a couple of remarks. The first one is that the frame in which we operate for novel foods is different from the frame for food additives, meaning that in the food additive regulations, there is a list, an a priori, let's say, list of foods in which food additives can be added, but this is the food additive regulation. For novel foods, the frame of our regulation gives us more freedom. In a sense, we don't have any predefined list of foods in our novel food regulation. However, having said this, as I mentioned before, since we share some principle with the food additives, meaning we are both interested in the chronic exposure. We also mention, we also recommend you to use both FAME tool or Diatex tool. So at the end of the day, if you decide to use the FAME tool to anticipate the intake of your novel food, at the end you will end up with categories which are, let's say, the very close category to the food additives regulation, and this will facilitate at the time of the authorization to end up with food categories also in the union list matching the food categories of the food additives. Then use of National Dietary Survey for food consumption. Let's put it in this way. I mean, the novel food is intended to be placed on the EU market. We EFSA, we need to evaluate whether the levels of your will be safe. We are using Diatex or FAME tool to anticipate the intake of your novel food in Europe. And these both, as I said before, the FAME tool and Diatex are based on national dietary surveys. So as a general principle, and it's mentioned also in the general principles in the guidance, if you want to deviate from the use of these tools, you can do it. You can use national dietary surveys as long as you justify why you are deviating, and then it will be up to the working group to decide whether the justification, I mean, the reason is appropriate, or otherwise we will come back to you asking to perform the exposure, the anticipated intake using the tools available. Then there was a question asking whether it could be possible to use a national dietary survey for the background diet. Yes, if you are interested in your novel food application to investigate the exposure to some components from the background diet, you can use national dietary surveys or the EFSA food consumption database or literature. Combine exposure of authorized uses and new intended uses. Here we have a specific question describing a very precise situation. Let's assume that we have a novel food authorized in the union list, and an applicant wants to extend the uses of that novel food, submitting an application and asking to add new foods to be included with the novel food. In this application, the applicant needs to present a combined exposure of the novel food coming from the authorized uses plus the new intended uses. Let's say, doing this exercise, there must be a difficulty, which is that the categories in the union list may not match 100% the categories in the FAME tool or in the Diatex, which are the tools used to perform the combined exposure. So the requester was asking whether it's possible to have a document done by EFSA to translate the food uses authorized in the union list into FAME tool or Diatex categories. Unfortunately, it's not feasible to prepare such a list a priori, a translation list. However, when an applicant is facing this situation, he needs to make his proposal on how to translate the foods uses authorized in the union list. In the union list, he needs to make his proposal, and then we will check when we scientific officer, we will start dealing with your application. Another option that you have on the table, especially for the post-transparency application, you may decide before submitting your application to apply for a pre-submission advice, for example. The last point relates to exposure to undesirable substances. Here, the requester was asking whether it's possible to have a list of minimum amount of undesirable substances. This is not feasible for two reasons. First of all, is that the minimum amount of an undesirable substance in a novel food depends on the exposure, on the intended uses of the novel food. And the undesirable substances in a novel food, they depend on the nature of the novel food. So it's really on a case-by-case basis. So what you have to do when you have to deal with undesirable substances is, first of all, to well characterize the novel food from a compositional point of view. As my colleague Hermolaus just mentioned in the previous slide, the importance of a well compositional characterization of your novel food in order to identify undesirable substances. So this is the first step. Then, based on the intended uses, as I said, maximum daily intake, if it's a food supplements, or based on the output of a FAME tool or Diatex, you need to take the highest 95th percentile of anticipated intake of the novel food. And then you need to consider the maximum amount of the undesirable substance in your novel food based on the compositional data, or the maximum limit if this substance is listed in the specifications. So having these two information, 95th percentile from the novel food, maximum amount of the undesirable substance in the novel food, you can derive and estimate the exposure to this undesirable substance from the novel food and compare this value with the reference value you have for this undesirable substance. So this was my last point. Thanks a lot for the questions received. And now I pass the floor to my colleague George Cass, who will guide you on the ADME. Bye.
Yeah, thank you very much, Manuela, and good morning or good afternoon for my side too. One of the issues, or one of the changes that the new guidance, the updated guidance document is highlighting is the need for biological and toxicological information. Now, this is not that we are asking for more or new information. It's just to make it simpler, make it easier to understand what is actually requested, because the same type of information was already requested in the previous guidance document, but not to the same level of description. So I will start with the ADME, and my colleague Wolfgang will then deal with the aspects of toxicology, those toxicological studies that are being requested. So to start with ADME, and here I try to summarize a bit the question that we have received. So first of all, the need for ADME studies. To be able to do a risk assessment, is really important to understand what is happening to the substance, to the novel food, as we're being exposed to it. First of all, is it broken down in the gastrointestinal tract to components that we know already, that are very well documented? For example, it could be a nutrient being produced that is very well documented, that we have all the information. A protein could be degraded completely to the individual amino acids. So this type of information is very important to know. It's also very important to know, should there be only partial degradation, but there's absorption, so the chemicals, the parts, the novel food or component of the novel food enters the body, it's very important also to understand what is the fate inside the body. Will it be metabolized? Could it be metabolized into a substance of concern, of health concern? So that is an aspect also that we need to know. And is it what is happening in terms of absorption if no absorption takes place? That is also a scenario to consider. And again, this will have implications on the toxicological data that are necessary, and the way the different triggers that are in the tox section that Wolfgang will explain to you are being activated. And finally, and perhaps that is perhaps a new aspect that really needs to be that we're emphasizing in the revised guidance, the updated guidance, is the aspect of human metabolism. It's very important to know that what is happening in experimental animals, so that's where we conduct our toxicological studies, that this is also, the metabolism is the same in humans, and that's a reason why we ask for comparative metabolism studies to really ensure that the metabolism in the animal is similar to what is happening in humans. And you will see also in the tiered approach on the graph on the right hand side that if this is different, then we have to go into immune studies straight away. Now this brings me also to the issue of determinants. So that has been a major question. What are the determinants that really should trigger the need for an in vivo study or in vivo studies for ADME? And here again, I would like to re-emphasize the importance really providing the compositional data, providing the specification is extremely important, and much of the information coming from that type of data will inform applicants on how far they need to go in the ADME studies. And it's also for that we use the tiered approach, because in the first tier it allows really applicants to have a look at what is already available, because it's not that we imposing studies in animals if the information, the critical information is already available in the literature, in reports, that the work has already been done. Also, whether the substance is a single substance, or food is a single substance, or whether it's a mixture, whether it's a mixture that has some elements, some components that could be critical in the safety assessment, or whether the components in fact are substances that are already known, let's say, to the body, that have been studied very well, and we know their biochemistry and their toxicology already. And I think it's also in this tiered approach important and to consider the nature of the novel food, because it's clear that it has to be a case by case. Some novel foods, it does not make sense to run them through a complete in vivo ADME study, nor often it is not possible to have this type of approach. We cannot run them through an in vivo study in animals. And this brings me to another question that was raised, and that is one of in vitro studies. So what can we do in terms of in vitro studies? Do we always need to go into in vivo studies? We put provisions in the revised guidance, the updated guidance, that certain questions can be answered using in vitro approaches. Of course, they often fall under what we call NAMs, so new approach methodology. So the emphasis is on the word new, and it's often the case that the approaches, that the assays that are available have not been validated yet, they have not been qualified yet, but we also made reference to some key developments that are currently ongoing at OECD level. There's, for example, in 2021, the PBK guidance document that OECD has published. We already give a lot of information on how to conduct this type of in vitro studies to look at absorption of chemicals. I will also recommend you have a look at the ICH document M12, which is currently under public consultation, that gives you some information on organotypics, 3D models that are available. And as time progresses, these models will become accepted and they can be used. Regarding animal studies, just to come back to animal studies and the issue of protocols, what protocols should be used? Of course, for animal studies, so toxicokinetic studies, there's the OECD test guideline 417 that should be followed. And for the in vitro, please refer to those documents that are described in the guidance document, and I've referred to the OECD PBK document and the ICH M12 document. So that is the issue of validation, and it's something I really need to emphasize. I think we're in the tradition of adopting new approach methodologies, trying to take us away a bit from animal experimentation, but we still are at the point where a lot of the in vitro tools that, and in vitro I'm talking also about in silico tools, not only about in vitro models, that these tools are currently still under development and need to be validated. So I think that's all from me. I think I covered all the key issues that have been identified and that you kindly sent to us. And I would like to give the word then to my colleague Wolfgang, who will be talking about the toxicological aspects. Thank you very much.
Thank you very much for the toxicology. Good introduction to George, because he has already answered a few questions related to NAMs and animal testing and product characterization. We got a lot of questions on the toxicological section, especially about the need of 90-day studies, about protocols, GLP, the triggers which require then in vivo studies, and triggers, questions on triggers which require higher tier toxicological studies. As George has already indicated, EFSA would really like to follow and apply more new methodologies which could replace in vivo testing. We have highlighted this in the general principle 11, promoting these issues, but at the same time we are also aware that these techniques need to be carefully validated. And that's still kind of a bottleneck, that's the truth, despite all our desire to reduce animal studies. So we would welcome more of these new methodologies, but we need also validation for this type of studies. So, and that's maybe the reason why we still rely largely for many novel food applications on this tier 1 toxicological data set, which are these in vitro micronucleus test, the in vitro mutation test with bacteria, the AIMS test, and a 90-day study. Now we got several questions related to novel foods, such as alternative proteins and fiber, whether these protocols, these in vitro genotoxicity tests and the 90-day study are really appropriate to investigate novel foods which are largely composed by macronutrients. And indeed, that's a critical point in many of our assessments. First of all, macronutrients, alternative proteins, are usually more bulky foods which are consumed to a larger extent than in certain ingredients, certain extracts, and it's very difficult to achieve to feed 100 times, 200 times higher dose levels to animals. So we also could not rely on these default factors we normally apply in the toxicological testing in results on 90-day studies or even chronic studies. So there's indeed limitations of these OECD protocols for 18 for the 90-day study and 471 and 487 for the in vitro genotoxicity test. And that requires indeed even more thorough analysis of the compositional data. Now we got 2 or 3 questions on whether a thorough compositional analysis could replace, mitigate, void the need for 90-day studies. And unfortunately, like in many other areas we have discussed already today, there is no yes or no answer, which could be applicable to all cases. It really depends on several issues, and that's, I'm coming now to point 1 on determinants for the need of in vivo studies, because the majority of in vivo studies we are assessing employing the most animals are the 90-day studies. So whether or not a 90-day study is needed or not, yeah. And there are some key elements which determine, which are often usually discussed among our experts, whether 90-day study is needed or not. We have tried to highlight this in the draft guidance. Maybe we should be even more explicit. The key determinants are, first of all, the source, the knowledge about the raw materials a novel food is produced. Similarly, the production process. So these 2 components, these 2 sets of data are absolutely essential, because everything that could end up in a novel food must come either from the source or during the production process. Also, things like production yield could be an important information. We had novel foods such as novel foods from tomatoes, from eggs membrane. I remember from milk we had a lot. Currently we're assessing one from a carrot. And if we have the information about the production yield, about how much of the raw material of a known source like a carrot and egg is needed to produce a maximum daily intake levels, and if this is reasonably consumed already as a source, there's also a strong argument against a 90-day study. Yeah? A thorough analysis and knowledge of the source and how much of this novel food could be consumed if it is related to the source. Yeah? The production process is obviously important. What other kind of components could end up in the novel food? Very essential, and which often triggers the need for a 90-day study, is if a large or a certain fraction of the novel food is not characterized. Usually we see this when we have plant extracts, especially from plants which have little or no history of human consumption. If there are extracts, ethanolic extracts, whatever kind of extracts, usually our experts demand for a 90-day study. And why? Because you can never characterize an extract from a plant, especially if the plant is not well known, to address all the components and the toxicity of all the components. Even there we demand thorough composition analysis, but here I see little chance of mitigating a 90-day study with thorough compositional analysis. Yeah. History of use is also determined, which is often used. It really depends on whether the source or the novel food as such has been already consumed in other countries. Depending on the data, on the country, on the system there, some weight could be given on these elements. Yeah? And at the end of the day, it's all these elements together which finally will result to the need for 90-day study, or in some situations our experts also will argue here it's not needed. Yeah? It's related to proteins. I think we never would ask for 90-day study for protein extract from alternative sources. It's never about the protein as such. It's always about byproducts coming from the production process or the source and the proportion of uncharacterized components. Yeah? Trigger for in vivo studies. I hope I have addressed this largely. What we want to avoid in our opinions and in the way we are assessing, and we hope that also our guidance is clear, that applicants would also jump directly to a 90-day study to mitigate the compositional data analysis. Yeah, so also the other way around should not be applied. That somebody says, okay, we have not done the proper analysis, but we have a 90-day study. That should also not be the case. Characterization, proper characterization, is always required, and in some cases it can mitigate, it can add to the evidence to convince our experts that a 90-day study is not needed. Yeah? We don't need a 90-day study to investigate fiber. It's also not about the macronutrients. It's always about the byproducts and the uncharacterized structure. And if there are components which have been identified, but where we do not have any kind of health-based guidance value on which we could argue these levels that could be consumed are safe. Yeah, related to this, we got also questions about other OECD protocols, such as on the an extended one generation reproductive toxicity study, when this is needed, and maybe could this be replaced with the shorter 4-2 OECD study, which is a combined repeated tox study and also reproductive toxicity study, but only with a shorter period of 28 days. We have hardly ever, I don't even recall that we had assessed the novel food dossier with a 4-2 combined study with a shorter 28-day repeated tox study and reprotox study. And why is that? Because I think whenever there is uncertainty with regards to reproductive toxicity, that the same issues will also trigger the need for a 90-day study. I don't think that there could be a situation where you have concerns regarding neurotoxicity, development or reprotox, and you don't have concerns with regards to subchronic or chronic toxicity. So if there are triggers that would demand for in vivo investigation of reproductivity and developmental toxicity, then I would rather go directly to the 443, the extended one generation reproductive toxicity study, which combines the 90-day study plus reproductive and developmental toxicity. That's especially saving animals if from the literature already you have indication that certain components in the novel food or the new component, the new substance as such, would bind to certain receptors on the nervous system, for example, or on the endocrine system. Things like this are triggering concerns with regards to reproduction and development. And in such a case, I think it would save time and money and animals if one would combine these two studies with an extended one generation reproductive developmental toxicity studies. But that requires thorough investigation of the available literature data. That's always important before performing any kind of animal studies or the complete toxicological approach. So that should always be, and I think it's also in a nice here in the slide, there's always the first step to take is the literature data. Then on the toxicity, we have the in vitro battery from the scientific committee guidance still from 2011, well established. Here sometimes we see applicants jump directly to in vivo studies. That's something we don't want to see, that animals are used before doing the 2 in vitro studies. Then we always go back to the applicant and tell them, please do first the 2 in vitro. Also because the animal studies are not always that sensitive. And for the repeated tox, following the decision we have discussed before on the determinants, whether or not a 90-day study is needed, yes or no. As an alternative to the 90-day study, one could really consider the extended one generation reproductive toxicity study, depending on the available information on a compound. Otherwise, the standard would be the 90-day study. We got the question, what triggers higher tier studies? Obviously, that's information findings from the literature or results from the tier 1 studies. Now I could name, of course, if in a subchronic toxicity studies we see some neurotox effects in the functional battery testing, for example, or if we see changes in the thyroid gland weight or reproductive organs on the uterus, testis, if there is something there in histology that could trigger then reproductive developmental studies. Very rarely, and actually I only recall one dossier where a subchronic toxicity study was followed up with a chronic study where there was some changes in the liver with liver cell hypertrophy that was followed up to investigate whether this could result to tumors, malignant or even benign one or even malignant one. But otherwise, usually we are, most of the time, let's say, 90-day studies are sufficient, and if they don't trigger concerns with regards to developmental reproductivity or carcinogenicity, then we are happy with a 90-day study as such. Yeah. But again, I would always carefully consider whether a 90-day study is needed. We had examples in the past already where applicants performed 90-day studies, and even the opinions we noted that such study was not needed, and that's also something that should not be done. And I also would like to stress is that we would go back anyway to compositional data, and often we do not ask in the first clock stop letter to go directly for 90-day study, especially when we have the feeling that thorough responses to the compositional data and the questions we had on the production process, if that could be answered well, our experts could live without a 90-day study. So in such cases, we always ask first to clarify the compositional data, to provide a full set and satisfactory information there before we ask for 90-day studies. In other cases where we know from the very beginning we will need a 90-day study anyway because it's an extract from a plant that has not been consumed as such with a considerable exposure, for example. Yeah. In such cases where our experts anticipate that they will need a 90-day study in any case, they would ask for that. Yeah. What other questions did we have? Yeah, on the OECD guidance, we also had a question whether a combination of toxicological studies which have been performed under GLB and other studies, non-GLB studies, but which have been performed under certain ISO or other national standards, whether this would also be acceptable. And I think here the regulation is very clear. The regulation it's the implementing rules 2479 from 2017 on Article 10 novel food applications. Basically, what is said there is that the safety, to demonstrate the safety of the novel food, those toxicological studies which are required to establish the safety, they must be performed under GLB. It does not mean that we would not accept non-GLB studies, but it means that for establishing safe human intake levels where we need animal studies or toxicological studies, these toxicological studies must be performed under GLB. Same applies for the genotoxicity test. So there's no margin there. Nevertheless, available studies from the literature, here we accept GLB, non-GLB studies as supplemental, as supporting evidence, but we would not establish health-based guidance values based on non-GLB studies. So I hope that is also clear. And also our experts accept non-GLB studies when it comes to studies like mode of action. But in order to establish safe human intake levels or to establish that the novel food is non-genotoxic, these studies must be performed under GLB. And under GLB, we also understand that such studies must have been performed in either OECD countries in labs which have been CLP certified for these type of tests, or in countries outside of the OECD if there has been a mutual agreement on this data with the OECD. Then these data are also accepted. In such case, these countries are also certified by their national GLP authorities. Yeah? So going through the questions, I think I'm done with the tox studies. What to test? Maybe what to test. Yeah, it's obviously important that the test material is very critical. Under GLP always requires sufficient information on the composition. So usually we get certificates of analysis of the batch that has been used in 90-day study or in the genotoxicity tests. These certificates are really important because no matter how good the study was performed, if this information is lacking, then we have a problem. So the test material must be really representative for the novel food. It's not necessarily always must be the novel food. It can be, for example, also pilot scale batch, which does not meet maybe 100% the specifications. Yeah, but there must be sufficient information there, especially on the presence of critical components from these test materials to allow our experts the conclusion that this test material is representative. When there are some protein levels or lipids or whatever do not completely comply with specification, that's the one thing that still may be acceptable, but there must be overall sufficient information that the test material was representative for the novel food. If not, then we had seen this, that 90-day studies could not be accepted. So I think I'm done with the talks to allow the other remaining sections, the few. Thank you. Sorry, and I give the hand to Ermalaus. Yeah.
Thank you a lot, Wolfgang. So now we are going to see what topics were raised by the registrants in the section of nutritional information. You can see here 4 main clusters. And when talking about novel foods in the European Union, we must remember 3 things. The novel food must be safe under the proposed conditions of use, must not be nutritionally disadvantageous, and also we need to remember that we don't assess the efficacy of these products and any eventual benefits. So the main thing that we, as it was illustrated before in the initial presentations, is that we try to elaborate further on the concept of nutritionally disadvantageous, because, and actually through your questions, we saw that there is the need to explain this concept. So actually, as I told you before, we try to follow a pragmatic approach that is all feasible for the applicants, is meaningful for the risk assessors, and also will be useful for the risk managers and policymakers afterwards. So nutrition disadvantageous means that the consumption of the product leads to an excess intake of nutrients, may lead to adverse effects linked to the nutritional status from the diet, or the nutrition disadvantageous concept can be linked to adversely affect the nutritional status of the consumers when it's replacing another food in our diet. And also the nutrition disadvantage can be linked to some cases where the novel food is proposed as the sole source of a specific macro or micronutrient in the diet, and this use as a sole source of a nutrient can lead to adversely affect the nutritional status of consumers. Regarding the anti-nutrients, which are the considerations that an applicant has to tackle when investigating the anti-nutrient content of a novel food. First of all, the anti-nutrients we must have in mind that there are components of the food that could interfere with the absorption of some nutrients. And in the guidance, we have tried to give some examples from previous risk assessment, previous published risk assessment. Of course, in the published literature through the EFSA journal, you can find even more examples on the anti-nutrient content of novel foods and addressing this issue in the risk assessment by EFSA. And it's always very useful to consider the source of the novel food when investigating the presence of anti-nutrients in the novel food. And we may have cases, for example, when we have a very well-known or consumed source already in our diet, but then the product, the novel food, is a concentrate, for example, an extract from the source, and the production process can lead to an excess to high levels of anti-nutrients in the final product. The third topic raised by the resistance was the protein digestibility. Actually, one of the sections of nutritional information is about about novel foods proposed as new sources of protein, and there we ask in this draft proposal for protein digestibility studies to inform further the investigation, let's say, of the concept of nutritionally disadvantageous. And this is, of course, not relevant for all the novel foods, but it's for those novel foods that can be a source of protein according to the existing EU regulatory framework, and also considering the proposed conditions of use, the proposed intake. For example, the applicant has to consider if the consumption of the novel food would lead to a substantial contribution to the dietary intake of protein. So the experts of the novel food working on the NDA panel, they propose to investigate the true ileal digestibility of the indispensable amino acids, and to do so, there are, of course, different ways, in ways that are mentioned by, let's say, international bodies. Of course, its approach may, let's say, lead to different results, and of course, the different approaches are not producing, let's say, results of the same value. And especially with the in vitro studies, the issue that came during our discussions here at EFSA is that there are many protocols under development to study the protein digestibility in vitro. However, there's not a plethora of protocols that have been validated. And as we said before, the validation of the protocol is something important for our risk assessments. So in the guidance, because the question was like, which protocol should I follow? Should I follow in vivo? Should I follow in vitro? So the guidance, let's say, leaves this choice to the applicant, leaves it open. However, with the note that the method, the approach used has to be validated. And there were also some questions specifically for certain protocols under development, and there, let's say, this concept would apply. And in our draft guidance, we cite the approach that was proposed by FAO, the Food and Agricultural Organization of the United Nations. And then in this document that we cite, the interested, the applicants, the manufacturers of the novel food, they can find the different methodologies available, and let's say they are even ranked in a way, let's say, of usefulness of the data that this method provides. And from our experience, also when we receive this type of studies, a major element is the calculation of the protein content, because in some cases, using, let's say, the conventional methods and the conversion factors also proposed by the existing EU regulation, we may have an overestimation or underestimation of the protein content in the novel food, of the true protein content. But this is something that should be considered when performing these studies, because this over or underestimation can, of course, affect the final result of the studies. And the last point was how to find what is an appropriate food comparator when let's say, preparing the nutritional information of the novel food, when, let's say, the applicants try to anticipate the nutritional assessment of the data that they're providing. So a very important element to consider is what the novel food may replace in our current diets. And also it's very important to consider, in case that we don't have, let's say, a direct replacement of another food, it's very important to consider the pattern, the dietary pattern that will, let's say, occur by introducing this novel food in the diet. And in these cases, for example, you can let's say, you can decide on possible food products that are already in the market being appropriate comparators for the novel food. And there are some cases where we had to investigate comparators. For example, there are some dossier. There was a dossier in the past and an assessment on the UV treated milk, and in this case, the comparator was the conventional milk, and the nutritional profile of both products was investigated and compared. So it's again on a case-by-case basis. But the very important thing to identify comparators is to consider what will be the role of the novel food in the European diet. And this is the main topics raised by the resistance in the nutritional information. So now I will give the floor back to Wolfgang to tackle the last section of the guidance about allergenicity.
Thank you very much, Amalaus. Allergenicity. We had got quite some questions here, as we expected, and in fact, personally expected even more questions and comments here, but sure we'll get some written in the written procedure in the public consultation. There were questions about the role of the literature data for this section, and obviously, like for other sections as well, it's an important element here as a first tier, I would say, especially for the category 3 and 4 of the groups. We have highlighted it now in the guidance documents. Now we have a bit more structured, I think, the guidance as it is now as compared to the present guidance. So this update indicates we have 4 groups of novel foods. Either there is no protein from the production process, the source, then obviously we have no concerns and no data requirements with regards to allergenicity. These are novel foods like mineral origin, synthetic products, or very highly purified substances where there is no source or potential residual of proteins. Then we have the category 2, which are anyway subject to mandatory labeling requirements. These are also easily to be addressed, no data requirements here, apart from the fact that we need the information from the production process that such sources are used for the production of the novel food. It gets a bit more demanding for category 3 and 4. Category 3 now is those novel foods where there's knowledge about allergenic proteins. And there obviously literature plays an important role, because in order to identify whether your novel food falls under category 3, novel food which may contain proteins with known allergenic potential, or category 4, novel foods which have proteins but where we have no clue about their allergenic potential. In order to determine whether your novel food falls under 3 or 4, you need to perform a literature search, comprehensive one, on your novel food. Yeah? It gets relatively easy for the category 3 when you have a novel food with a known allergen. there we basically ask to put together from the literature information about the severity of symptoms, about the prevalence, obviously always only if available. So we don't ask you to generate this data, but to take them from the literature. And third, on the potency, which is often not available. Yeah. And the other requirements for this category 3 known allergens is then to try to identify them in the novel food if they are there, and to quantify them, the known allergen. And basically that's it. Why to quantify? Because that may contribute, that's an information that we can use to compare the exposure, how much of this known allergen is consumed with the novel food as compared to the source, let's say. Yeah. That's information that would contribute to the overall assessment. Yeah. It gets a bit more complicated than for the category 4, where the literature does not give you information about known allergens. But obviously you need to perform this literature search in order to come to this conclusion that your novel food falls under category 4. That leads me to the next set of questions about in silico methods and how to follow up for potential cross-allergenicity and how to address more complex novel foods. Yeah? Category 4, as we said, concern novel foods with proteins of unknown allergenic potential. And there we have, let's say, 2 subcategories. One is novel foods which contain basically one or very few proteins, which can be well characterized and analyzed one by one. And then the more complex novel foods, cell culture, protein extracts from plants, or whole biomass from fungi, where we have a lot of proteins, how to investigate these. And I think, and here I think we need to further explore how to make the guidance more clear. I understand from the questions we got here, because our aim is to a kind of a tiered approach. First, the literature search, and second, then to do in silico methods to analyze homologies of the proteins. That's relatively easy if you have only 1 or 2. It gets a bit more complex if you have a complex mixture whole food, but it's doable nowadays with proteomics techniques and bioinformatics. And I think we noted somewhere in the section 4 that it's not the de novo sensitization we aim for, because there are no methods and validated tests to investigate the de novo sensitization potential, but it is more targeted to cross-allergenicity. And when you apply proteomics and homology analysis, we have referred to certain thresholds, and if they are met or exceeded, that could demand to a next higher tier study, or next step, let's say, which is an IgE specific immunoassay, such as an ELISA or Western blot, to investigate whether the protein for which you got a hit with regards to a homology to a known allergen, that these proteins then are investigated with human IgE specific antibodies, ideally, I think in the guidance we set from patients, with this known allergenicity, not allergenicity, with the known allergic reactions, allergy against the known allergen. We got one question, what to do if such human sera are not available? Then obviously you cannot do it. We had an example in the past on rapeseed proteins. Rapeseed brassicacea come from the same family like mustard, which is one of the 14 allergens listed in the annex of the Consumer Information Regulation. And in this dossier, the applicant told us, look, we tried everything to get serum from patients suffering of mustard allergenicity, allergy, and we could not obtain such serum, and we accept it. If it's not available, it's not available. Yeah. But in such a case, then we would expect that the applicant tells us that he tried, what kind of institutions he has approached, and that nothing was available. We also must admit, or I would like to explain that several of the requirements here now goes a bit beyond on what we have asked so far until now. Because allergenicity is part of the risk assessment and safety assessment of novel foods, and we know since beginning of EFSA that the European Commission and member states, they have never refused authorization because of an allergenicity of a novel food or its proteins. And the question is usually among member states and Commission only about labeling yes or no. And especially last years, it became more and more evident that member states are very reluctant to introduce new labeling warning against potential allergens coming from novel foods. And only if the data indicates that a novel food really contains an allergen that could trigger more severe reactions with a higher prevalence and even with a lower potency, these elements, then member states are more inclined to impose labeling measures. So a reasonable question, and I got this sense from the comments we got already now, is why we even asking data what is beyond than needed by member states and Commission for their labeling decision, basically. Yeah? And we have drafted the current guidance with the idea, or the current draft as it is now, with the idea that this additional knowledge about the allergenicity potential of novel foods is reasonably to be asked by applicant, because nevertheless, whether it's used or not used by risk managers, it's still part of the risk assessment, and it's legitimate to inform science, public, about available information such as cross-allergenicity in vitro, even if we cannot make a conclusion that the novel food is likely to trigger reactions in human. Yeah. So this type of evidence is basically, the idea is to collect, to impose to applicants to generate a certain data set, which will be then available for the public, whether or not this will be considered by risk managers. And we know that some of the questions and requirements we have now in the guidance will, at the end of the day, not be considered by risk managers. No? I also must admit, and I think the allergenicity section is maybe the section that still may undergo further discussion within our experts, with the Commission. We also await, of course, the input from the public consultation. And especially maybe in the category 4, those novel foods with unknown allergenic potential, there may be some revisions. Yeah. So we are really appreciating any kind of comments you may have to give also in the written public consultation. What other questions we have here? Dose response, if this refers to the threshold or potency. You may know that neither EFSA nor we are aware about any kind of thresholds which have been established. We know that some allergens have higher potency, others less. The only thing what we could do is to compare the exposure of an allergen in a novel food as compared to what we would consume from the source. That's the only thing maybe we could do related to quantity of an allergen, but it's basically impossible for the very most proteins to do anything about dose response, and we would not demand any kind of testing or analysis there, because we know it's the results will not be very useful. But of course, if there's information from literature about the potency, this information should be in the dossier. Intolerances, we got one question was whether allergenicity should be understood in the context of the consumer information regulation with these 14 allergens, which include also lactose and sulfides, sulfur dioxin, which are not allergens but substances which could trigger intolerances. How to investigate these? We have nothing related here in the guidance. Obviously, if we are aware about the compositional analysis about the presence of a food that is known to trigger intolerances, we would make note in our opinions, and the applicant should refer to it and maybe even do some quantitative estimate how much of this component would be consumed, lactose or sulfur components or even histamine, at the high intake estimates. But at least to our knowledge, there's no validated, generally accepted study to investigate for intolerances, unless maybe you do a huge human study. But that's something not realistic to be asked in these novel food assessments, because you would really need a lot of subjects, and that's something we cannot do. Major minor allergens, I think that relates that includes some managerial decisions. We know that the WHO, they consider the severity of the clinical reactions, the prevalence and the potency, whether an allergen is, let's say, more minor or major. That's scientifically sound, of course, but at the end of the day, that's a managerial decision whether you consider something as major or minor, and therefore we don't have this concept as such, these terms as such. But we are asking if there are data on the prevalence, on the severity, and on the potency in the literature, but we do not use these terms as such. Yeah, and on the tiered approach, I've a bit already highlighted it on the homology analysis of the amino acids. When certain thresholds are met, a follow-up is required with an IgE binding test. Maybe we should make it a bit more, emphasize this tiered approach for both single proteins and complex mixtures. That the main target of this approach is to investigate cross-allergenicity rather than de novo sensitization. That could be maybe a bit more clear in the guidance. Thank you from my side. I think I'm done with the allergenicity. Yeah. And I think it's Anna. Thank you.
Yes, it is me. And I think we're reaching the end, which I'm sure everybody will appreciate, because it's quite late in the afternoon. I don't even dare to think at what time there is in other parts of the world. So a very, very big thank you to everybody that stays with us. An impressive number, I have to say. I'm really impressed. And I hope this session has been useful to the ones attending. And again, my very big thank you to all the ones that have submitted questions and made this session possible. Of course, thank you to all my colleagues and all the team that has been behind the computers trying to make this webinar possible. What else to say? So I think the most important thing is that the written public consultation on the draft guidance is open until the 14th of April. So please read it, comment, criticize, everything that you can do, and we would very much appreciate. What we want is a guidance that is fit for purpose, that can help applicants, that can help assessors do the best work possible, and in the minimum time possible. So it's very much built and designed with that in mind, to make sure that our dossiers are as complete as possible, so we can also have the risk assessment completed timely. What else? Maybe just a couple of words to say that there are also other 2 guidances under public consultation. This session was specifically looking at the novel food guidance, but there's also the micronutrient sources guidance document, which is currently under consultation, and the traditional food guidance also for consultation. So if it's not too much to ask, we would also appreciate comments on those 2 documents. Now, the recordings and all the presentations, as I said, will be available on EFSA's website in the coming days, probably next week. And the other thing is, please take a few moments to just complete the satisfaction survey that you will receive by email. It's, as I said, a very useful tool for us to improve. And I think this is all. Have a wonderful evening. Thank you from all of us. There's a couple of links here. We're always available to answer questions and comment and provide explanations when it is. There is a Ask EFSA, which I invite you to use when you send your questions to EFSA. And have a wonderful evening. Thank you all.